In 2013, once again an animal influenza A virus has crossed the species barrier to cause an appreciable number of human cases. Are you aware about the epidemicity of avian influenza A H7 and 9? Yeah, it was first reported in China in March 2013, and currently, China is experiencing the fifth epidemic wave of this influenza. 100,000 of duck, birds, and chicken have died due to this virus, and they have been killed in order to curb its spreading. According to WHO, the total cumulative number of human infection with H7N9 since 2013 is nearly 1,600 cases, and 39% of it confirmed that with this virus. At first, this virus only circulated in avian species only. Unfortunately, the first human infection due to this virus in 2013 was starting point of a new flu virus worrisome. The success of this virus adaptability in human system is questioned, and the mechanism of this virus to keep surviving and become robust should be clarified.
The viral genome exists in single-stranded negative sense RNA, and these RNA are segmented into 8 pieces. In influenza virus, there are 2 RNA segments that are expressed in both spliced and unspliced form, which are an X segment and M segment. And now we are focusing on this regulation of splicing of NS mRNA segment. The NS segment expresses 2 viral proteins, non-structural protein NS1 and nuclear export protein NEP. During infection, NS1 protein functions to alter mRNA processing, leading to inhibition of host antiviral immunity.
In the splicing process, the NS1 protein interacts with the host splicing regulator SF2 in the nucleus. The NS1 protein regulates the host splicing process by interacting with small nuclear RNA to prevent the formation of spliceosome complexes and subsequently blocks nucleocytoplasmic transport of host mRNAs to intervene with expression of genes required for host antiviral function.
This interaction also contributes to the regulation of NEP mRNA splicing.
NEP is regulated to remain at low levels to avoid negative effects on viral transcription during the early phase of infection. In other words, the level and timing of NEP expression significantly affects viral replication efficiency. However, in the later stage of infection, NEP is required for coordination of nuclear export of viral ribonucleoprotein.
So to sum up, as the production of NS1 and NEP proteins are mutually exclusive, the regulation of splicing of NS mRNA segment has an impact on the viral replication efficiency.
This can be explained by the role of G540A. G540A substitutes itself in ESE site, which is found in NS segment of H7N9 virus. This gene allows virus replication in mammalian cells but retains its replicative ability in avian cells. Furthermore, NS1 protein interaction with SF2 enhances NEP NS1 mRNA splicing. This splicing process relies greatly on host machinery. The splicing process is initiated by the binding of SF2 ASF to ASE site to produce NS1 mRNA. The NS1 then binds to suppress the splicing process. But the NS1 mRNA that is already produced is then spliced to produce NEP mRNA, which is essential for virus replication. This uniqueness of interaction leads to the abilities of H7N9 virus.
In this study, Huang and his co-workers tells us about G540A substitution in H9N2 that provide the internal gene for H7N9. However, there are many factors that contribute to G540A substitution in H7N9. So, this study should include what is the factors and what are their mechanism that contribute to G540A substitution. Second, other than H7N9 infection, people in China are also affected with H10N8 in 2013 and H5N6 in 2014. Other scientists said that both of these viruses contain G540A substitution, and there are no cases reported that this virus infect people without G540A substitution. Thus, Huang and his co-workers should include the adaptation of G540A substitution in both of these viruses and make comparison to H7N9 virus.
First of all, it allows us to understand how do these viruses evolve in order to infect other organisms, cross-species transmission. I mean, different virus has different biochemical structure, right? As for this study, this avian virus relies on host splicing machinery for processing of viral mRNAs. Secondly, this finding actually references that this virus still retains the ability to infect birds even though they have changed their protein structure to infect humans. If we can observe and gain much information about how do they adapt to this, how do they have this kind of ability, maybe we can crack down all the viral infections for good.